Today, we gather to celebrate outstanding scholarship and to recognize a scientist whose work has opened new frontiers in our understanding of life at the cellular level. It is a great honor for Kyoto University to present the Kyoto University Lectureship Award in Life Science and Medicine 2025 to Dr. Clifford P. Brown-Wine.
It has indeed been a sort of exciting journey to launch the selection process for the inaugural laureate of the Kyoto University Lectureship Award on Life Sciences or Medicine. As the inaugural laureate of this award for his work on the discovery that liquid-liquid phase separation serves as a fundamental principle of intracellular spatial organization.
Please join me in a round of applause as we welcome our laureate, Professor Brownwing, to the stage to accept the award.
Okay, in recognition of outstanding contributions to the advancement of science, Kyoto University hereby confers the Kyoto University Lectureship Awards 2025 in Life Sciences or Medicine upon Clifford P. Brown-Wine for his discoveries of liquid-liquid phase separation as a fundamental principle of cellular organization. Kyoto, Japan, April 16, 2026, President of Kyoto University, Minato. Congratulations. Oh. Beautiful. Okay.
Thank you very much. Please join me once again for a warm round of applause.
Yeah, I've prepared some formal remarks that I'll make later to express my gratitude, but for now I'll just say this is really an incredible honor, and certainly to be the inaugural recipient of this award I think is very special. So I thank you so much. This is really quite an incredible honor. I want to start out by saying what I view as the fundamental problem in cell biology today, which is the question of how the components of living cells come together, how they assemble or really self-assemble into cells and tissues and organisms. This led to this picture wherein the segregation of these P granules, the way that they move over to the right side of the embryo, is a kind of phase transition, what we refer to as liquid-liquid phase separation. Science is a team sport, and this is certainly another message I think for the trainees would be important is that everything we do that's interesting, every paper that I've ever written has many co-authors and is highly collaborative and involves many brains and ideas and creativity. I've been incredibly fortunate to have many mentors, colleagues and trainees. This is an incomplete list. I can't show them all and name them all, but just gives you a sense of the many people that I've been incredibly fortunate to interact with and learn from. I just feel very blessed to be in this profession and to have learned from all these people and really the recognition that I'm so grateful for receiving today from Kyoto University should be viewed as a recognition of this group and the field that has formed around these ideas.
You should specifically suppress some kind of LPS. The other ones leave intact.
Yeah.
Do you have any strategy to specifically suppress?
So after quite a bit of hesitation and discussion and some hesitation, we decided to invite the people from different departments of the university.
So these two papers marked a turning point in my research, and that possibility deeply excited me. And then I immediately sent the email to the CRIF. And then we met, fortunately, in Tokyo sooner later. So I think they're students here, so what I want to say is that please don't hesitate to send the email to fantastic scientists. So, and then more later, the CRIF accepted me as a postdoc, and then we tackled like a central question in the LLPS field.
So this is a conclusion. The commutative forms a stable condensate during transient input, and commutative condensate alters receptor distribution of PSD, and commutative condensate captures and accumulates various synaptic proteins into the stimulated synapses via CBM. And take-home message for the neuroscience, commutative condensate acts as a bioelement of the memory via persistently reconstructing PSD. And for the general, this is the title. Inducible condensates provide a tunable platform for the spatiotemporal protein integration. And so let's condensate our LLP's research potential.
The B4 mutant shows impaired HPI foci fusion, indicating that B4 segment is involved in the regulation of proper heterochromatin organization in vivo. We combine the result of NMR, SEC-SAX, and the CGMD-SAX analysis to construct the following model of LLPS. At low concentration, interaction between the phosphorylated tail and the basic segments are mainly intramolecular, resulting in compact conformation.
So I will not go into the physics of separation here, but the key point is that pyrimidine can self-organize through simple molecular interaction between riboscore and EPIC1. So this discovery changed how we think about the pyrimidine. It was no longer the strange body in the chloroplast, but it became something we could study in terms of assembly or dynamics or control. So, and one thing that makes the pyrimidine especially interesting is its unique structure, architecture. Actually, the circle membrane, which has a membrane, penetrates into pyrimidine, even though the pyrimidine itself has no membrane. That is very interesting.
Finally, I would like to express my sincere gratitude to all those who have made this event possible, including our partners, sponsors, and colleagues. The Kyoto University Lectureship Award has taken its first step today. We are committed to further developing this initiative as a platform that connects the world's leading researchers and nurtures the next generation of scientific leaders. Once again, please join me in congratulating Dr. Brangwyn. Thank you. Thank you very much.
Inaugural laureate of the Kyoto University Lectureship Award, and you guys have put me on a pedestal. But remember, I'm just bumbling along like anybody else. And you know, I'm trying to learn as I go, and if you're having doubt and you're wondering and you're having failure, don't worry, because I have it too, and really anybody does. They just don't usually advertise these failures and anxieties and insecurities. But I'm here to tell you, everybody's got them. You know, the imposter syndrome. Does everybody know about imposter syndrome? You know, academic career is so interesting. It's very, very multidimensional. there's always different problems and different, as we've discussed. It's not about doing problem sets or solving equations or things. Yes, that's great. Good. And if you're good at that, good for you. But this job is a whole vast space of interesting parts to it. And so I think people can bring a lot to the table along many of the different dimensions. So.
先生のそのキャリアパスとかこの時に何を考えておられたかっていうのがそう詳細に分かったので若手研究者学生の我々としてはすごくためになったというか
クロスティング・ザ・フィールドっておっしゃってましたけど、ちょっと違うジャンル分野に行ってるんで、そこ難しさもあるんですけど飽きませんねあるっていうところで、そういうので結構勇気づけられたっていうところがあります。
Just say, well, we had a wonderful event this morning and got to interact with the students, and I tried to share in my talk just a little bit about some of my experiences in the adventure of science, and really grateful for the opportunity to be here and speak with the students. They then gave some lightning talks, and I was very impressed with all the things that are happening here at Kyoto University, and I learned a lot and very exciting things. So yeah, thank you for having me.
Thank you.